sign up for normal today.
It's not as good as it is.
Okay.
From what I've heard, it's like a lot of
I've heard it's a lot of math, yes. Our roommate is taking the regular.
Ooh, that's why I became a biologist. I was running from that. Okay, today we're going to talk about nucleosome assembly. It's a little bit out of order. Probably should have come before talking about the events at the telomere. But I discovered when I looked at the module 4 that there were a lot of things in setting it up on UF Online that the course designers, helpers, did not include. It's not their fault, but anyway, so that whole module 4 is under extensive revision. So it really doesn't pay to plan ahead and be well prepared, because I'm going to be changing it like crazy. We're not going to spend the whole rest of the time on the pre-initiation complex of transcription. We're going to have a chance to talk about some epigenetics and X chromosomes and calico cats and RNA polymerases 4 and 5 and heterochromatin and lots of really fun stuff. In addition to more than you ever wanted to know about the pre-initiation complex of Paul 2. So here we go, nucleosome assembly. This has been a part of the course, so this is not new. I've updated just a little bit this morning and removed the blue and all that fun stuff. So there are two basic pathways to assembly. Those pathways which are dependent on DNA replication. And they have proteins called chaperones that assist in the removal of histones from the DNA just before it's being replicated, and then the reassembly of a combination of old and new nucleosomes back onto the newly replicated DNA. So this puts a big demand on histone production, right? All of a sudden now you've got twice as much DNA. That means you need twice as many nucleosomes all of a sudden. Well, nucleosomes are comprised of histones, and histones are very basic proteins. If you have a lot of these just kind of hanging around with nothing to do until the DNA gets replicated, it can be very disruptive to a cell to have all this basic charge around. So one way to store proteins that are highly charged is to bind them to something that's highly charged with the opposite charge. And some of these chaperone proteins are acidic, as you might expect. What would they be mimicking? Why are histones so basic in the first place? Because what they naturally bind is acidic. DNA itself. DNA, the phosphodiester backbone, is very negatively charged. So histones neutralize that, so you can condense it way down tens of thousands, or at least 10,000 times. So it's quite logical that proteins that assist in the folding and assembly of nucleosomes in a way mimic DNA, at least as far as their charge. And so you have these stages of assembly, and you can see here there are other slides that show this, but you start with, it almost recapitulates evolution. We have the archeal situation here. The first thing you do is you assemble these H3H4 dimers into tetramers. This is as far as it goes in archaea, and then we carry one step further, which is the addition of 2 copies of H2A and H2B, one on the top, one on the bottom. And each of these steps is chaperoned. Here you see the chaperone called N1, and there's one over here called NP. N1 is one you should I'll put on the keywords list. So replication dependent, replication independent. What is replication? What would be an example? One of the major examples would be transcription. As the RNA polymerase moves along, at some point you have to bump the nucleosome off. And this can happen in vitro. RNA polymerase doesn't need any assistance. But in vivo, it's facilitated and made much easier and smoother with chaperones. So the first category will be the replication dependent. This is the pathway that follows along closely behind the RNA polymerase. And the key word here to know is CAF1. CAF1 assembles the nucleosomes after replication has occurred, taking a combination of new and old. And here you see CAF1 participating here. And sometimes it also participates in repair operations. So this replication dependent can be expanded to include repair. So it's more properly called replication and repair dependent pathway. You'll notice on here that PCNA has been labeled. PCNA, remember, is the clamp that is responsible for processivity of the replication enzyme, the DNA polymerase. It's a 3 subunit ring. In eukaryotes, a 2 subunit ring. It would be the beta clamp in bacteria. But PCNA has affinity for CAF1. This is how CAF1 is able to follow along so closely behind the replication fork. It's a part of the complex. It's attached to the tail end, the back end of the polymerase, through the clamp. Okay, CAF1 is responsible for putting together the H3, H4, and I am not sure whether it actually does H2A, 2B or not. Often you'll have multiple chaperones, but at a minimum, it handles histones 3 and 4. Notice also what's here. There's another factor which seems to be sort of ahead of the pathway. These are sometimes called escort pathways. You escort the chaperones to their final destination, handing off from one complex to another. This ASF1 is not specific to replication dependent pathways. This participates in both replication independent and dependent. In one case, it hands off the partial histone to CAF1. In doing transcription, it will hand it off to the other partner, which we haven't talked about, but which will often appear on exam, called HIRA, which we'll get to in just a minute. This is a better, more detailed picture of what's going on. You have the tetramer comprised of 2 copies of 3 and 4. Can you recognize those? 4 would be green, 3 would be in blue. See the praying mantises there doing their little dance? The blue would represent 3. So this is a tetramer. Here you see the tails with the modifications. And then this is joined to preformed dimers to end up with the octamer. Here you see the participation of ASF1 and CAF1 in, this would be replication dependent nucleosome assembly. Remember, ASF1 is not specific. It participates in both pathways. In this particular case, it is handing off H3, H4 to CAF1, which is in turn being pulled along behind the replicase. So you're seeing a combination of old and new nucleosomes being assembled on both strands. It's a mixture. Is that a good thing? You want to keep it pure so the new daughter cell has all the brand new histones, still shiny, got the full warranty? Or do you want to mix it up? Well, first of all, who's the daughter, who's the mother? I mean, they're both. The mother becomes both her daughters. There's a Greek myth there somewhere, I don't know. But it's fortunate that it's a combination in the reassembly, because that is how the epigenetic message or pattern is learned by the daughter cells. These patterns of epigenetic modification of the histone code spread themselves once they get a seed. So this is how the new nucleosomes become marked like the old nucleosomes, because they are heterodispersed. Here you see apparently ASF1 doing lagging strand and CAF predominantly doing leading strand. This is misleading. This figure does not mean to imply that. They both participate in the nucleosome assembly on both strands. How about genes that are being actively transcribed? You can't see that very well at the moment. I'm going to outline it here in the next little, I think, well, yeah, there. This is SV40, a very robust mammalian primate virus. And you can see there the circular DNA genome of the virus. And coming out here, it's hard to see now, but that's the RNA that's being transcribed from one very strong promoter, making a very long transcript, which will be cut up later. So the answer is yes. See the beads on a string here? Even something extremely active in transcription is expected to be coded with nucleosomes, with an exception we're going to see in a minute. What would you think the exception would be? Something that's even more heavily transcribed than a virulent virus.
The ribosomal.
Ribosomal's right, and we talked about that a little bit. There's so many RNA polymerases packed on the ribosomal cistrons that you do the theoretical calculations and you see there's just not enough room there for nucleosomes also, for how large it's spread out. So that's sort of an exception. Here you see that exception, the ribosomal so-called tree. And once again, pay attention to this formation here. The RNA polymerases would represent the black nodes at the base of each of these little branches, and the branches, of course, represent the transcript, in this case, the ribosomal. And you see each of those starts with very small RNA, ends up with very large RNA, small RNA, large RNA. So you could just look at this and tell me the direction of transcription, right? You go from small RNA, and the more you transcribe, the longer the RNA gets. So direction of transcription would be that way, and this way, and that way. It says length of the so-called tree here is 85%. The predicted length implies that the gene is almost completely opened up in its most exposed state. There's not even beads on a string here. Whereas you can see transcription by Paul II, you do have nucleosomes. So it just depends. In most cases, with that one exception, the ribosomal cistrons, you still have B's on a string. Histone octamers, are they displaced, bumped aside, or does the polymerase just go round and round and round, then go to the next one, go round and round and round, and go to the next one? Turns out they are displaced from the DNA and have to reassemble. And since we brought up the topic of transcription here, you can imagine that this is a transition into replication independent nucleosome assembly. Transcription is the best example of that. Octamers reassociate with DNA soon as the polymerase has passed. Is it a fair contest when you have a nucleosome and you have a little bitty mouse of a polymerase sneaking up and knocking on the door, excuse me, could you get out of my way? Or is it some big, hulking RNA polymerase kicking things out of its way as it goes? Who would win this contest? It turns out that RNA polymerase is a little larger than a nucleosome. Nucleosome is pretty big, right? RNA polymerase with its 12 subunits is even larger, about 500 kilobases. This is this in vitro experiment that shows that histone octamer is displaced from DNA and then rebinds. This is from the textbook. RNA polymerase can transcribe through a nucleosome in vitro if unassisted. However, in vivo, it does have chaperones to aid. And of course, you're going to need to know the name of that chaperone. So here's another keyword. I think that facilitates chromatin transcription. FAC. Fact. Not alt fact, but fact. This is the complex that moves ahead of the RNA polymerase and participates in the disassembly of the nucleosome. It has to work pretty quick, right? And specifically, its job is to remove the caps, the H2AB and H2AB dimers that are on the top and bottom of the nucleosome as the first step of disassembly. Fact. Transcription, you know, that repair, I think the book said that, but I'm going to cross that out with an X when I do my next revision here. CAF1 participates in repair, and I think this slide's in error. Very often in nucleosome assembly, it's a chance to slip in your own message, like put an advertisement in there. And there's a variant histone which is used for that purpose, to signal what kind of region of the chromatin this is. That's histone H3. That shows variants that reflect sometimes the cell cycle stage, but quite often whether or not this region is being heavily transcribed or not. Because FACT, in association with ASF1 participates in both pathways. It sort of is a pipeline, taking the nucleosomes, the new ones and old ones, the parts, and handing them either to FACT or handing them to CAF. There's one other thing it hands it to, which is HIRA, which surely there's a slide on it somewhere. So the histone variants. Histone 3.3 is the one that is slipped in there when the nucleosome is reassembled. Turns out histone variant 3.3 has lysine 56 acetylated, which reduces its basic charge as a histone, which means it doesn't bind the DNA as tightly. Now, lysine 56 is not located on the tail. It's actually located in the histone fold, that region of the histone that actually interacts with DNA in the wrapping. And so this makes sense in terms of our model about how things work, that it slips in a variant of histone 3 that makes it easier to disassemble that nucleosome next time around. Histone variant 3.3. It's put there after repair also. I don't know the logic behind that. Okay, so we've talked about how they're disassembled. How are they reassembled? They're reassembled using this HIRA. And I don't know what that stands for. I can look that up, but I don't know. But HIRA is sometimes used on exams for, can you imagine, a fill in the blank? Really? You expect us to remember that? HIRA, H-I-R-A? Now you'll never forget it. HIRA. HIRA. My dear HIRA. It has been so long since I've seen you last. Okay, go on, there's nothing on me. Okay. Centromeric DNA replicates early and uses, here we go, another histone 3 variant. Histone 3 variant at the centromere, the CSE4P or the SNPA in humans. So this is a common theme that histone 3 is used to alter nucleosome structure for one reason or another. Okay, so that's the end of assembly. Now we have a few more topics and then we can get into the fundamentals of the transcription complex. So as we sort of ease into that, let's talk about regulation. We've looked at how genes are well, we haven't looked at how genes do anything yet, so let's begin to look at how genes work. We talked about the loops, these large, 90 kilobase domains that seem to be attached to the nuclear matrix, and the suggestion has been that you regulate the entire loop as one unit in some sense. You open it all up, the entire loop is opened at one time, and that's kind of a gross level of regulation. There's also the problem of how tightly can you pack genes? Can two promoters be right beside each other, one going one direction, one going the other? Well, you see that all the time. If they are together like that, do they cross-regulate each other? Or if they don't, how do they know which one they should be regulating? So it turns out all of the above is possible. Sometimes they cross-regulate, and other times there are boundaries to their influence. These boundaries to influence are things that are still a hot topic of discussion. Boundaries. Insulators is the name of the protein that would bind to some of these boundaries. So if a gene is very highly induced, will neighboring genes be active? Is this a lug wrench? Maybe. What stops heterochromatin from extended into regions of the genome that contain genes that should be actively expressed? What keeps heterochromatin from spreading? So we'll begin to get just take a crack the door on that big topic and talk about some of the proteins that do this. The proteins bind to these boundaries, whatever they are. So you can, in your mind, you could construct a boundary two ways, right? You could have your insulator protein recognize a DNA sequence that's between influence domains. I must bind there, for I am an insulator. I must stop cross-regulatory traffic. Or the insulator could bind to modifications on the histone tails, epigenetically determined boundaries. Probably both of these hypothetical modes work. We know there are actual sequences in the DNA, but one could easily speculate that it's all of the above. Enhancer activates a protein, a promoter protein, but can be blocked by having an insulator between them. What's an enhancer? You don't know that yet. A promoter is the sequences, the controlling sequences that signify the start of transcription and regulate that initiation process. And these are clustered very close to the start of transcription. Just like in a bacteria, the promoter is a very defined number of bases from nucleotide 1 of the transcript. We call that the core promoter. There are a collection of protein and big complexes that recognize the core promoter and guide the RNA polymerase, inform the RNA polymerase as to where it should start transcribing, because the RNA polymerase will start most any place if you don't give it explicit instructions. So the core promoter, and then in eukaryotes, especially in metazoans, you have clusters of control elements called enhancers. These are really made up of multiple submodules where different kinds of transactivators, sometimes repressors, will bind. And these are usually tissue specific in the kind of expression they confer on a promoter. Now, in the normal world, the enhancer, the closer the enhancer is to the promoter, the stronger the effect, just by probability. Just things are just bumping in the dark, the more chance it can bump into core or basal transcription factors that are assembled here, the greater chance it has to recruit and influence. But it turns out metazoans have made a big jump. They left plants way behind. They are able to regulate their gene expression hundreds of kilobases distal, away from the promoter. This is something a bacteria couldn't even conceive of, and it turns out, and I've only learned this in the last 6 months. I spent my entire career on plants saying they were so marvelous. I could do everything and they wouldn't scream when I cut their heads off. And now I see they aren't the same as animals. They can't do this, I don't think. I'd like to do the experiments to test it, but we'll go into what this might be. How do metazoans exert influence from great distances? And why would they need to do that? The metazoan organism is a much more complicated collection of tissues and cell types and has a much more intricate and precisely timed developmental sequence as it goes from zygote to mature organism. Perhaps that has driven the need to step up to another level in terms of their potential for regulation. And that is what I assume has happened. So we'll see, what was it that provided that breakthrough? And would I get arrested if I had an undergraduate cloning team in my lab who introduced this to plants? Is that illegal in Florida? Probably. I don't know. I shouldn't think these thoughts, but that's what tenure's about. It protects me from saying crazy things and getting fired. At least that's what they keep telling me. I don't know. We may test it. Insulators can block activation and repression on promoters on either side of them or both. How do they do that? We're just beginning to appreciate and learn how this works. It can block the spread of heterochromatin. How does that work? Here's a picture of the Drosophila again. This is a polytene chromosome. This is close to 1,000 linear copies of the DNA laying side by side, perfectly lined up. So it diffracts light. That's why you see the bands. You see band and inner band. And you see these regions that seem to be kind of puffed out, called puffs. This is one of the more prominent puffs that appears, and this occurs if you increase the temperature by 10 degrees centigrade very rapidly. You induce the heat shock response, you get these puffs, and this is where they were first discovered by an Italian researcher. These puffs are flanked by insulators, we now know. And here they are. And this is something you need to recognize on exam. These little SCS and SCS prime, these are the, says the chromatin structures, but there's an actual sequence in the DNA, 350, 200 base pairs. So this is not an epigenetically determined site for where you put an insulator. This is actually encoded in the DNA, and there must be a DNA recognition protein. The insulator itself must recognize where to bind. This is in Drosophila, and you see it flanks that 87A7 puff. Why would you want to do that? Why is that a good place to put an insulator? Because these genes are really, really super transcribed. You might want to limit their influence just to that region. SCS and SCS prime. The protein that binds it, there at the SCS and SCS prime, is called BEAF, and I don't think that that term is on the exam anywhere. Just insulator binding proteins are all you're going to need to know. I mean, you don't have to know every single protein in the human genome. Or maybe. We'll do that for summer C. So this is an example where they have fluorescently tagged, and you can see that the insulator, the BEAF site is it's actually both in bands and in between bands. Protein binding is localized in interband regions. It says interband. Okay, we'll go with that. We'll say interband. RNA polymerase do not co-occupy the same regions. The insulator is green, fluorescently tagged, and the antibody it recognizes Pol2 is red, and you can see, as you might expect, that regions that block the spread would not be intermingled with regions that have RNA polymerase for active transcription. So what happens if you mutate the proteins that bind this BEAF protein, insulator binding proteins? There are a whole family of proteins that act as insulators. Well, at the top you can see that it actually messes with the structure of the chromosome itself. All this polytene, nice parallel layers of newly replicated genome side by side. Because transcription now can go everywhere, you can disrupt the structure of the polytene chromosome itself. In addition, you can have drastic effects on various developmental processes. Here's one particular insulator protein disrupted. You see the wild type Drosophila I, and here you see its disrupted form. You can see some breakdown in the normal pattern. Wild type, disrupted insulator, disrupted. Here's more drastic. This is really a drastic phenotype. So these things appear to be real, and we're just beginning to appreciate their role. The end of nucleosome assembly. That wasn't so bad, was it? Stuff probably you haven't heard. You've heard around the edges of it, probably. Did you cover this in biochemistry? You didn't? Yes. Okay, we got a few more minutes. Let's go into something really fascinating. Let me see here. Where would it be? In the beginning. And this next 3 PowerPoints part, what's it going? You think that's a mistake, don't you? Maybe it is. Can't say at the moment. So it looks like my little last minute getting things together here was kind of time not well spent. So I'm assuming that there are other parts here, even though this is labeled 1, eukaryotic transcription. Well, I hate it when I do this to me. I know, it's the blue background. So let me give you a little preface of where we're going here. What does a eukaryotic Pol-2 promoter look like? What does Pol-2 do? Transcribes genes that encode proteins. So it has a transcript that must undergo extensive processing. You start with 100,000 very large transcripts that have mostly intron. Introns have to be cut out. You have to stick a poly A tail, add it to the 3' end, and you have to cut and make a 3' end because the polymerase is not going to fall off and terminate. You have to cut and then add 200 to 400 A's on the end, and then at the 5' end, you have to add an extra nucleotide. A 7-methyl G has to be stuck onto the end. And remember why that's there? It's to communicate to the ribosome that this is the end of a messenger RNA. This is not the end of a ribosomal RNA. This is not a fragment of RNA being degraded. This is a messenger RNA because that's what the ribosome is looking for in a eukaryote. It is not looking for a Shan-Delgarno. There is no Shan-Delgarno. There's some translation optimization sequences there, the Kozak consensus and so forth, which is there sometimes and sometimes not. But there's no ribosome binding site. When a eukaryotic ribosome finds that cap, that 5-methyl cap, it begins to scan. And the very first ATG it comes to, it assumes that's where I should start translation. Now about 20% of the time, and in some of your genome projects or gene structure projects, you will find a little short open reading frame that the first methionine that it comes to actually is sort of the warm-up band. The real translation product is at the second methionine. And that first one may have 3 or 4 amino acids in it and sort of hands it off to the second one. We don't understand how that works or why it should be. It probably occurs because it doesn't seem to disrupt anything. But it's not always the first. But if you're taking a bacterial gene and trying to engineer it into a plant or a nematode or a kitty cat, you have to remove any ATGs in the untranslated leader. Otherwise, the eukaryotic ribosome will start transcription right there. So that's one thing. Pol-2 is what we're going to be talking about. It expresses all the protein encoding genes. So what does the promoter look like? I've already talked about there being a core sequence. At the core, you get a series of complexes called basal transcription factors. They bind to every core promoter, regardless of whether it's a cell type, whether it's brain or liver or spleen or skin. Every promoter has a core region close to the start of transcription that attracts a pre-initiation complex. It begins to nucleate at that start of transcription, and the proteins that assemble there are used in all genes. Like RNA polymerase has to assemble there. Well, there are a series of basal transcription factors that are not gene specific that nucleate at the core. And then you have more tissue and timing developmentally specific binding sites for transactivators. So you have 2 general things. You have basal transcription factors, which assemble at the core promoter, and then you have transactivators and repressors. Transactivators recognize the DNA of the element, just like in bacteria is what you'd expect. The transcriptional control protein, whether it's positive or negative, recognizes an element in the promoter. Well, eukaryotes have really complicated it. There are a class of proteins that recognize elements in the promoter, but do nothing except do that, and then they in turn attract other proteins, which in turn attract other proteins, which really have the business end of whether you're going to shut a gene down or activate it. And those other proteins that do not know where to bind are called co-something, co-activators or co-repressors. The repressor itself recognizes the DNA element. The transactivator itself recognizes the DNA element. Other proteins that are then cluster at that spot are labeled co-something, co-activators, co-repressors. So let's look at one of these basal transcription factors. This was the first to be characterized extensively, and they all have letters. TF2, 2 is Roman numeral 2 for RNA polymerase 2. These are basal transcription factors that associate specifically with RNA polymerase 2. And they have letters like TF for transcription factor 2, TF2A, TF2B. There's not a C, but there's a D, there's an E, an F, an H, a K, and that's all they are. But they were named according to how they eluted off a hydroxyapatite column a long time ago, some graduate students' midnight experience. A, B, D, F. So this particular one is D, TF2D, and it has a central, very famous central subunit. And then it has its friends. The central famous subunit is Tata binding protein. It just goes by initials, it's so famous. TBP. TBP, oh yeah, TBP. TBP, right here, Tata binding protein, actually is the only one here, well, not really. TBP knows what a promoter looks like. There's a Tata in the promoter, just like in bacteria. It's a little bit different, but it's essentially the same sequence. And the big distinction is location. In bacteria, this is located approximately -10, right, from the start of transcription. In a eukaryote, it's never located at -10. It's always further back from the action. It's usually from about 20, 24, sometimes 21, but people like to say it's at -30. It's quite often at -30, but between 21 to 30 is where you're going to be looking for it. If it's not there and it says Tata, don't believe your lying eyes. It's not a functional promoter element. It's just another AT-rich sequence. So when you're trying to identify promoters and all this, and it's a eukaryote, if it's not in the right location, it's not functional. Because this forms a complex which determines where the polymerase must start. It can't help itself. The polymerase is going to have to start about 30 nucleotides downstream from it. So it's called a commitment factor. I hate that word, not the factor word. This TBP is a commitment factor for all of the polymerases, Pol 1, 2, and 3. Wow. When it's in association with Pol 2, it has its own little special friends that only know that it thinks TBP is like married to Pol 2. It doesn't know it's got 2 other families on the other side of town. It goes together with another group when it's with Paul 1, and another group entirely when it's with Paul 3. TBP. Who invented TBP, that rascal? Mama Archaea. That goes all the way back to Mama Archaea. She decided that this was how it should be. You know, the Egyptian god, what is it, with the M? Is it Mu? The mother goddess that represents the heavens. She bends over, arching over in the sky. Sort of like TBP there. Oh, you could say it's just a lima bean. So what are the transcription factors that associate? Whoops. TBP associated factors. Oh, that's hard to remember. TBP, they're called TAFs. Now, if you look at the annotation in the gene banks, they'll go RNA polymerase II associated transcription factor. Really, all they need to say is TAF1, TAF2, TAF9, or something like that, because that's the real name. They were annotated before people knew how to talk about them. So you have, 10 to 14 TAFs that are associated with TBP when TBP is with Paul 2. Oh, it says 9 to 14, I guess. Look how large it is. It's larger than RNA polymerase. Now you know who's the mama. Nucleosome 300, RNA polymerase, the big bully, is 500, but TF2D is 800 kilobases. Whew. How does it work? What does it do? Functions. It recognizes the core promoter. And you're going, yeah, it's TBP, recognizes TATA. Most of your genes have no TATA. Did they tell you that in biochemistry? They were afraid to tell you that. You wouldn't come to class ever again. I've already gotten over that. Most of them don't. So what does TBP do? The promoters for Paul 1 and 3 don't have a TATA. So it's used to being left alone. There must be other proteins here that know what a promoter looks like if it has no TATA. Indeed, that's the case. It's like your Garmin. You put in those little information disc, and that's where the brains are. These guys are going, okay, there's not going to be a TATA here, but we know what to look for. And they know other core elements in this promoter to look for. So one of its functions, the TF2D. TF2D is the entire complex, TBP plus the TAFs. One of it is to recognize the core promoter. It's a target for recruitment by transactivator proteins. This means if a transactivator bound to an enhancer, the transactivator has an affinity domain called an activation domain that is sticky. It has affinity for the various TAFs. And as it's kind of fishing like an anglerfish at the bottom of the sea, and it runs across TF2D, it'll stick on one of these subunits and reel it on in. Make it tethered so that now it pretty much has to bind and start the nucleation process. In fact, this is the first thing to arrive at the core promoter, TF2D. It's a target for recruitment by transactivators, and when it gets here, it now begins to fish. It has affinity for the other basal transcription factors that must also nucleate here at the promoter. You end up with more than 50 proteins in complexes and subcomplexes, all congregating here at the start of transcription. Enzymatic activities. One of the TAFs, only one so far, has enzymatic activity. The TAFs are numbered according to their size. The largest is TAF1. And so they are numbered sequentially down to the smallest being TAF14. TAF1, the largest, has 3 enzymatic activities. What kind of thing would you want to do there with enzymes? Maybe modify some histones. There is a HAT, a histone acetylation enzymatic activity there. A couple more we'll talk about in just a minute. How much time do we have? 0 time? I'm just getting wound up. Okay. I can hardly wait till Thursday, right?